So hemoglobin consists of two parts, which is the heme, a non-protein molecule, and the globin or the polypeptide. So for the formation of the heme, it takes place in both the mitochondrion and some of its processing takes place in the cytoplasm. So first, in the mitochondrion, the succinyl-CoA formed in the Krebs cycle binds with the glycine in order to form a pyrrole molecule. This molecule will convert to delta-aminolevulinic acid, which is the first byproduct of heme synthesis. This first byproduct will go to the cytoplasm and two molecules of this will be converted to porphobinylogen via the enzyme ALA dehydratase. Next, this molecule will be converted to uroporphobinylogen 1 by the enzyme synthase. Afterwards, this molecule will be converted to uroporphobinylogen 3 by the enzyme co-synthase, and an enzyme called decarboxylase will convert this molecule to coproporphyrinogen 3. This molecule will go back to the mitochondrion and will be oxidized by the enzyme oxidase to form protoporphyrinogen 9 and will be further oxidized to form protoporphyrin 9. Afterwards, an enzyme called ferrochelatase will help bind the ferrous ion to the protoporphyrin 9 in order to form a fully functioning heme. So for the globin synthesis, it starts with the primary molecule wherein various amino acids will join together in order to form a polypeptide via the condensation reaction. This would result to a water molecule product. Next, the heme, which is made here, will go to the polypeptide and will attach itself specifically to the nitrogen positive atom of the histidine imidazole. Next, this polypeptide with the heme bends and folds to form either an alpha helix or a beta pleated sheet for a more stable molecule. This is an example of a secondary structure of alpha helix which contains 72 to 89 residues. For the tertiary structure, this molecule will continue to bend in space in order to form a more stable molecule. And the heme here is an important factor in shaping the protein and connecting to certain histidine residues in the polypeptide, creating a 3D structure. And lastly, for the quaternary structure, separate tertiary structure, specifically 2 alpha chains and 2 beta chains, will combine in order to form 4 globins and 4 hemes, making the fully functioning hemoglobin. This hemoglobin can now collect oxygen, specifically 4 oxygen molecules needed by the organism.